for 6 weeks at different stages of pathology, and to assess neurofibrillary pathology and memory. So we did this at 4 different stages in the disease and asked what the effect of suppression was on neurofibrillary tangle pathology. The 2 time points that I want you to focus on are here, number 1 and number 2. You can see when the transgene was suppressed at 2.5 months of age, the tangles went down. But when the transgene was suppressed at 4 months of age, the tangles didn't go down. In fact, they kept increasing. So what that means is that the progression of tau pathology becomes independent of the levels of transgene expression sometime between 2.5 and 4 months of age. Okay, so once this tangle pathology is triggered, and if it progresses beyond a certain point, then even suppressing 90% of the expression of the mutant tau has no effect on the accumulation of the tangles. What happens to memory? So, if you keep the transgene on during the whole time period from 2.5 to 4.5, 7, 9.5 months of age, and you measure memory function, which we do using a Morris water maze, and the higher the number, the better, and 25% is random. You can see that the memory function declines and stays poor. If you turn off tau at this stage, when we have mature tangles, and actually we do have neurodegeneration by stereological estimates done in Brad Hyman's lab, these animals here have only 50% of the CA1 neurons in the hippocampus. What happens then is that if you turn it off briefly, the memory function improves, and it stays improved even for extended periods of time. So what this tells us is that despite the fact that the neurofibrillary tangles keep going up during this time, memory function is getting better. And as it turns out, you might notice that in the previous slide, you actually see more tangles here in the suppressed condition than in the non-suppressed, and that's because there are actually more neurons here in which to develop tangles. So that's the cause of that slight increase. So from these conclusions, we can see that tau-induced memory impairment is reversible, that the memory function improves regardless of neurofibrillary tangle pathology. So it doesn't look to me like neurofibrillary tangles are the cause of memory problems, so they correlate very well in humans. That it associates neurofibrillary tangles as a cause of memory impairment, and it supports the hypothesis that there is some form of soluble dysfunctional tau species that precedes structural changes, like the tangles or even the neuronal loss, which can exert deleterious effects on neuronal function. And we don't know what this species is. We are calling it tau star. It's an unknown, and Shelley Zhao in my lab is the project leader for discovering tau star. Now, let's look at this model of A-beta amyloidosis. It's actually a model that I know much more about because we generated it in 1996. At first, we were quite excited because there were subtle memory deficits in amyloid plaques, and I thought we had generated a model of Alzheimer's disease, but these mice do not develop neurofibrillary tangles, a very important feature of Alzheimer's disease, and they have no neurodegeneration to speak of. So they're missing 2 of the really important late-stage features of the disease, and I come to the conclusion, and this is my assumption when I work with these mice, that they do not have Alzheimer's disease. Instead, they represent elderly individuals who are at high risk for developing disease, and they may have some subtle memory problems, just as people who get older have a harder time in the name face-bearing tasks. But these animals, in my opinion, do not have Alzheimer's disease. So what can we learn from these animals? Well, first of all, we gave these mice out, and there are several other mice that have been made. And at least for the 2.76 mouse, there are over 300 publications showing that one form of treatment or other can help these mice. And I listed some of them here. You can see blueberries, Cabernet Sauvignon, as well as real drugs like ibuprofen, naproxen. And none of these, with the possible exception of naproxen, has really been able to make at least a big impact, in my opinion, on the human disease. Even a paper, which is my favorite, where they engineered potatoes to express a beta protein, and they fed the potatoes to the mice, and the mice got better. So that was the best one, I thought. Anyway, when we first looked at these mice, we wanted to know whether the plaques had something to do with the memory problem. And what we found, Marcus Westerman and my lab found, was that the memory problem preceded the plaque deposition by 6 months. So it did not seem as though the plaques were causing a memory problem. So then we wondered, is it A-beta related at all? And Linda Cotillion in my lab showed, well, it seems to be, because if you passively give A-beta antibodies, you can reverse the existing memory deficits. And this result was replicated in a different model, PD-ATP. So it suggested that brain dysfunction in these mice and plaque pathology were mediated by different pathways. So this raises a question and generates a hypothesis. What role does the amyloid beta protein play in memory loss? It's not through plaques. What is it through? We hypothesized that since it was occurring independently of neuronal or synapse loss, we hypothesized the existence of a species which we just called A-beta star.